All right, good morning, ladies and gentlemen. We're going to go ahead and talk today about translation, which is also known as protein synthesis. I really prefer the phrase protein synthesis because that phrase makes total sense to me. You're building proteins. Translation, I have to sit there and go, what does that mean again? But anyway, so translation is the third part of this central dogma prezi that we've been looking at. So I'm going to go ahead and click on that. Give me a moment here and we'll get started. All right. There we go. Okay, translation is the last part. It's where we take our RNA and it's brought to a ribosome and it's turned into a developing protein, also known as a polypeptide. You might remember from biochemistry. So this image we've seen before. This is sort of showing you the whole thing. So there's the DNA double strand. In the nucleus, it's separated. The RNA polymerase read it and made a copy of one side called the template strand. And this is what it built. It built an mRNA strand, the messenger RNA. Now, the messenger in a eukaryotic cell is going to leave the nucleus. It's going to head to a ribosome, and it's going to tell the ribosome what protein to build by telling it what amino acids to put together. The amino acids bonded together make our polypeptide or our protein. All right, so let's look at a little bit more detail. So first of all, let's talk about the ribosomes. Interestingly, ribosomes are the only non-membrane bound organelle. They're made of RNA, so they're also called rRNA. And this is where translation or protein synthesis is going to be taking place. All cells everywhere on the planet, bacteria through eukaryotic, all have ribosomes that are interpreting what the mRNA has to say. So we're going to learn a lot more about the ribosomes right now. This is a computer-generated model. With some of these models, and you're going to see a video later, they try to show like each individual atom or molecule of the molecule. Oh my gosh, that's hard to say. Anyway, and so they're a little bit more complex. And so then we ask artists to simplify those really complicated shapes. And so you get these sort of cartoon shapes. And so the cartoon shapes are super helpful for training our brains what we're looking at. And then we can go ahead and look at the real molecular shapes and then go, ooh, that's what the cartoon was about. Okay, so things you need to know about a ribosome. It has what's called a small subunit, which is shown in yellow here, and a large subunit, which is shown in blue here. It reads the mRNA, so this represents the mRNA right here, and there are three areas within the ribosome where the trucks, the tRNAs, are going to pull up, and that'll make sense in a minute. And then the polypeptide chain or the growing protein starts to form right inside of it. So let's take a look at the cartoon version. Let me get this out of the way. So here's our cartoon version. We've got our small subunit here. We've got the large subunit here. And you need to know these three sites, the A site, the P site, and the E site. Now, when you're just staring at it, you're like, why didn't KJ say EPA? Because we usually read right to left. Well, in this case, the mRNA is entering from the right and it's traveling this direction. So it's going to encounter the A site first, then it'll move to the P site, and then it will move to the E site. So as it's traveling through, it's actually going APE, so think of APE instead of EPA, the Environmental Protection Agency. All right, so the A subunit is where the tRNA is going to first join, and I know you don't know what a tRNA is, but tRNAs I often call the trucks because it starts with a TR. So they're trucks. What do trucks do? They carry supplies. So in the cell, the trucks are carrying amino acids. So the trucks first pull up into the A site, then it dumps off its whatever product it's carrying, its amino acid in this case, and then the whole ribosome shifts 3 letters on the mRNA. 3 letters on the mRNA are called a codon. It's like a secret code. So the whole ribosome shifts over 3 letters, and the truck now is in the P site, which is called the peptidyl tRNA binding site, which just means that the amino acids are going to bond. So whatever amino acid was there before is going to bond to the new amino acid. And we start getting a polypeptide that starts forming out of this exit tunnel. Then the ribosome shifts again, and that causes the the now empty tRNA to eject. So that's how it leaves. It exits right here. It is now an empty tRNA, and it gets recycled. Coolly enough, everything in the cell gets recycled. So now it is an empty truck that is just going to pick up the same product. So in the real world, a truck that's for, say, that carries plumbing supplies, it's not going to go pick up carpeting. It's gonna, once it's gone to a construction site and it's dropped off its plumbing supplies, it's gonna go pick up plumbing supplies again if it's a plumber's truck. And so it's not gonna go back and pick up something different. So our trucks in this analogy are always picking up the exact same amino acid and recycling over and over and over again. All right, and then you can kind of see it more visually here. Here's the mRNA traveling through the ribosome. The ribosome is constantly shifting, and so the trucks are moving from one of the sites, from the A site, then to the P site, then to the E site, where they exit. And you see the amino acids join together to form that polypeptide. All right, so now we're going to give you a little bit more detail about the whole process. Oh, I forgot, we have to tell you about tRNA in a little bit more detail also. So these are the trucks. There are a bajillion different ways to draw all of these things. And so because they're super complex molecules, the artists draw them different ways. So this drawing in the middle, letter B, is more realistic for what the actual molecule looks like in a three-dimensional shape. It's a single-stranded bunch of RNA. It's single-sided. It's not like DNA that's double-sided, but it has a tendency to twist and fold on top of itself because some of the nitrogen bases are attracted to each other, so they form hydrogen bonds with one another. So it has a complex shape. There are very important parts to this shape, though, that you need to understand. Now, the first drawing is if you kind of took it and smashed it flat, and now it's a two-dimensional shape. It, of course, never actually looks like this. And then in our textbook, to simplify the drawings and to make them not so that you can focus on the right part and not focus on all the details, I've never seen them drawn like this before, but they're going to be drawn like a little bent tube. Don't ask me why, but that's the shape that they've chosen for this book, and that's totally fine. So this is the two-dimensional shape, and I want to just show you the really important parts of the two-dimensional shape. The first important part is up at the top where it's called the amino acid attachment site. So this is right here. It doesn't have an amino acid right now, but where my arrow is circling, that's where the amino acid would be carried. On this diagram, on the three-dimensional diagram, it's off to the right. It's over here. That's where it's going to be carried. The other part that's super important, and it occurs to me, I forgot to say something on the previous slide. So give me a second. We're going to go back to this slide. I mentioned it, but I want to be really clear about it. So on the mRNA, the ribosome reads the mRNA 3 letters at a time. So as it's shifting, it shifts on the mRNA 3 letters at a time. It's constantly moving at 3 letters at a time. So those 3 letters are called a codon. It's the secret code. And the codon is it's called a codon because it is a code that tells the cell which amino acids need to be brought in which order in order to do what the DNA wants it to do. So that's called a codon. Well, how do the trucks know what the codon is? Well, it's not that they know what the codon is. It's that they, at the bottom, have 3 opposite letters, 3 opposite nucleotides called the anti-codon. And these molecules are attracted to each other. Opposites attract. So if if the codon on the mRNA if it read UUC, then this tRNA would be attracted to it, and it would come by carrying its amino acid, and it would drop off its amino acid. So the anticodon is the opposite 3 letters. And remember that I use the letter U because in RNA there are no T's. T's are more stable, more energy intensive. That's why they think RNA doesn't have T's and DNA does. But regardless, remember that RNA is always going to be using U's. All right, I think that's enough about tRNA. That gives us the basic idea. One thing that the textbook does go into a little bit of detail is just how do the trucks recycle and get their amino acids. So it turns out that there are 20 different enzymes. Remember, enzymes are involved in absolutely everything. Anything that happens in the cell, there are enzymes involved. You haven't even learned a billionth of all the different enzymes that are working and doing things in your cells. So there are 20 different amino acyl tRNA synthetase enzymes. There's 20 of them because there are 20 amino acids that your body is using to make your DNA, to make your proteins. So just like there are 26 letters of the alphabet, and we use those letters to make our words, there are 20 amino acids, and this should be a review from your biochemistry unit. There are 20 amino acids that we put together to make our proteins. So there are 20 enzymes that attach the amino acid to the truck. So there are 20 of these enzymes that attach the amino acid to the truck, and they only fit the right amino acid. So this is a different amino acid won't fit in here. So that's why there's 20 of them, is that there's exactly the right number. And then it says here, a tRNA with an attached amino acid is said to be charged. So you could call it the amino acyl tRNA if it has an amino acid on it, or you could call it charged tRNA. I like to just think of them as trucks that are carrying building supplies, but whatever. You choose the fancy word that works for you. All right, and then hopefully you've already seen the genetic code here, but it's super important that you take a minute and really analyze what you're looking at. So these are 3 letters and which amino acid they're coding for. Now, look at the table for a second. If you ignored on the outside everything, you'd probably be fine. So here are the 3 letters. Here's U, 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 and it codes for the amino acid phenylalanine. Here's UUA, and it codes for the amino acid leucine. Notice that there is some repetition, meaning that there's more than one codon that codes for one amino acid. For example, for proline, there are 4 different codons that code for proline. What is very important that you understand is that the first 2 letters are always the same. It is only the 3rd letter that changes sometimes, and that is referred to as wobble. And I'm not going to go into the details of it, but just trust me that wobble allows several different codons to code for the same amino acid. All right. Something that's really cool about the genetic code is that it is universal across all domains of life. Bacteria use A, U, C, G, and T's, and DNA and complex eukaryotic cells do that. So what they're showing you in this diagram, which I think is pretty darn cool, we have a pig that has been given by scientists, a little bit scary, but cool at the same time, a protein called a gene called GFP from a jellyfish that allows it to glow. So under black light, this pig's nose and other features glow because it's been given a jellyfish gene. How was that possible? Jellyfish use the same types of letters that pigs do and that humans do. And so we can insert genes from other organisms. Is that always a good idea? Absolutely not. But it is possible. In this case, really cool. This is a tobacco plant that has been given the luciferase gene, which I love that name. It's wickedly cool. Anyway, and so that's what allows fireflies to glow. And so this tobacco plant can glow because crazy scientists put that gene in them. All right. So, I thought it was going to close in on this. So things to understand about this genetic code over here. The triplet code, mRNA is read in units of 3 bases that are called codons. Remember, the trucks have the anti-codons. So I sometimes emphasize the T in anti to remember that they're on the trucks, on the tRNAs. There are 64 possible codons for just 20 possible amino acids. The code is redundant and unambiguous. And what that means is that for example, redundant means that there are 4 different possible codons just for proline, so it's redundant, it's repeating. However, it is unambiguous. Always, CCU always means proline. CCU will never mean histine or serine or phenylalanine. It always means proline, so it's very specific. And then the other thing to point out on this is that the codes here also include a start codon. Every single mRNA begins with methionine and the 3 letters AUG. It's called the start codon, and every single amino acid, every single mRNA starts with AUG. Then, importantly, there are stop signals. So 3 of the 64 codons are actually stop signals that tell the ribosome, okay, this is the end, and now you're done, and the ribosome can go away, and the polypeptide has been built. I'm going to skip this video. Feel free to watch these videos at any time that you would like to. All right, now we're going to go into the detail of exactly how the protein gets built. So initiation is the first part. Let me back up so you can see it. Initiation, elongation, and termination are the 3 steps. So with initiation, get my photo out of the way here, the mRNA attaches to the small ribosomal subunit. So here's the small subunit, and the mRNA is attached to it. Methionine, remember we said, is always the first one. It's brought to the start codon. So the start codon is in red here, and if you could read it, if they made it big, it would be AUG. So the opposite 3 letters on the truck, oh, they did make it big. Now I see it. So UAC, that's the anti-codon on the truck. It's attracted to the opposite 3 letters, so that truck pulls up and it drops off its methionine. The ribosome large subunit then clicks on, and it says the start codon is in the P site right at the beginning. This is called the translation initiation complex. I hate that word. Do we really need a name for this? But apparently we need a name for this. So translation initiation complex. I'm okay just saying, oh, it's the ribosome with the mRNA going through it, but whatever. They give a big fancy name for it. All right, so the tRNA binding at the ribosome is mediated by an anticodon loop in the tRNA molecule. All right, so I'm going to move on to the next. Oops, I did that wrong. There we go. Next section. So keep going. Elongation is the next section. So now we're going to actually start building our polypeptide. We're going to make it longer. So we start, if you take a look at the whole diagram, and this diagram is in your textbook, and we're going to jump to the upper right-hand corner here. Give me just a second. So the next codon, remember that we have, it's not showing you the first step, but in the first step, it was just methionine. Now they've shown you that a whole bunch have already come through. But after the ribosome has shifted over by 3, the next codon determines the next amino acid to be brought to the ribosome. So whatever these 3 letters are, that's going to determine which of the trucks that has that anticodon, which truck is going to come up and drop off its amino acid. The incoming charged tRNA enters at the A site. This is where it enters. With a little bit of GTP used, remember GTP is like ATP. It speeds up the process, makes it a little bit faster, more efficient, whatever. And so now it has docked, the truck has docked, it is dropping off its amino acid, and now the amino acid is going to bond to the existing polypeptide chain, all these amino acids. Remember from biochemistry that the bond that forms between 2 amino acids, remember that there's a carboxyl acid group at the end of this one, and there's an amine group on the other. That's where the name amino acid comes from. Through dehydration synthesis, and they're not showing you that water gets released, but it does. So through dehydration synthesis, this one gets bonded to this one. So they actually scooch over. Let me get my picture out of the way. The growing polypeptide is transferred to the new tRNA molecule, and a peptide bond is formed. And now, oops, sorry, I hate that about this. There we go. The ribosome shifts. It's also called translocating. Do we need to say those words? I don't know. Shifts. The tRNA with the polypeptide is now in the P site. So the polypeptide was attached over here, but now it's going to shift. And so now notice the truck with the polypeptide growing is in the P site. A little bit of ATP or GTP gets used for this process also. And now the uncharged, remember we called trucks tRNAs that have their amino acids, we're calling them charged trucks. Now it's uncharged, it's empty, it doesn't have its product anymore. So the uncharged amino acid is now in the E site and it gets ejected. And then the whole process happens over again. The next codon is now available in the A site, so notice that it's available for the next incoming charged tRNA. The ribosome is ready for the next amino acyl tRNA, which is the charged tRNA. It's ready for a charged tRNA. All right, we are done with that. That was elongation. And now termination. Just how does this whole process end? So all the way at the end of the mRNA, it's going to encounter a stop codon. And there are 3 stop codons, so any of the 3 stop codons will work. it releases, a release factor binds to the A site and it triggers the whole thing to come apart. The polypeptide chain is released and the ribosome disassembles. And now we have our protein. All right, so differences between I am going to show you this video. So this video, I'll try not to talk over it too much. This video shows you now the more realistic. It's going to show you every single little atom instead of just the artist's impression. I'll let the narrator talk, but actually I'm going to pause it. Okay, so what you're seeing right here, this purple area, go away. This purple area right here, that's the nucleus. It looks like the surface of the moon. And then all these little holes, those are nuclear pores. They look like craters on the moon. And what's coming out of it right now is an mRNA. So we're seeing the mRNA coming out of the whole process. All right, I will turn her volume up.
Protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell.
The purple thing is the ribosome.
These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life.
So the red, those are a bunch of amino acids put together forming a protein.
Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs.
I'm going to pause it there. So if you remember the three-dimensional structure of the trucks, the tRNAs, they kind of bent over to the side. So that's what this is. This is the three-dimensional structure. And in this rendition, the amino acids are sort of a glowing red ball. It's like a little Rudolph the Red-Nosed Reindeer nose or something. So little nose on the end of them, that's the amino acid. On this end, why does that keep popping up? On this end over here, that's where the anticodon is going to be, and that's where the opposite three letters are going to be.
That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of three letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are three locations inside the ribosome, designated the A site, the P site, and the E site.
Ape.
The addition of each amino acid is a three-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
All right, we'll go ahead and stop there. And next. Go away. I don't know how to make it go away.
The flesh and blood.
Nope, didn't want that. Moving on. Oh my gosh.
Code is
I'm gonna have
quite
to hit
literally
escape
turned
to make it go
into
away.
flesh
Give me
and
a
blood.
moment. I don't know why it wouldn't jump on for me. So I'm going to move ahead. And what is the problem? And I can't pause this video. Sorry, you guys. Finally, there we go. All right, moving on from that video. Oh my gosh, that was such an exciting video. Okay, not really. So differences between prokaryotes and eukaryotes, actually quite interesting differences. Remember that prokaryotes don't have a nucleus, so their lifespans are amazing. Remember that they do all their living and then they reproduce within 20 minutes. We have a reproductive cycle that takes us 20 years to reproduce, sometimes more than that. So prokaryotes don't have a nucleus, so they don't need the mRNA to leave the nucleus, and they could do everything all at one time, and it's really pretty cool. So since prokaryotes do not have a nucleus, transcription and translation can be coupled. They can happen simultaneously. So they have something that are called polyribosomes. Polyribosomes are the simultaneous translation of a transcript, even while that transcript is still being made. What? Take a look at the diagrams. They'll make sense to you. So if I look, this is what I think is the better diagram right here. Here, the blue line represents DNA. Look at the guitar picks that are reading the DNA. Those are RNA polymerase. And so they're reading the one side of the DNA, they're reading it, and they're making mRNA. And so that's what the red ribbon is that's coming out of each of these. So it's all happening simultaneously, which is amazing efficiency and speed. And then because it doesn't have to leave a nucleus, right away the ribosomes clamp on and start building the polypeptides, start building the proteins. And a whole bunch of them can be building the proteins at the same time. So that's referred to as a polyribosome, when they have a whole bunch of ribosomes that are working at the same time. We don't have that in eukaryotes. Eukaryotes cannot couple transcription and translation. We don't have polyribosomes. We also need to target where the polypeptides that are forming, where they're going to go in the cell, because the cell is so enormous. And that's where we have to have things like, is that the next slide? It is the next slide. That's where we have to have things like the endoplasmic reticulum as our highway system that's going to move the polypeptides to the correct location in the cell. So let me get out of the way again. So signal peptides, a small signal peptide sequence on polypeptides that need to be made at the endoplasmic reticulum. So some polypeptides, not all, some polypeptides are going to, right at the beginning, have a signal of 3, I just made up that number, but certain number of amino acids that trigger a particular protein called the SRP protein. The SRP protein recognizes the signal, and then it brings the whole ribosome and the growing polypeptide to the endoplasmic reticulum. Remember, this is the part of the ER that looks rough because it has ribosomes joined to it. So the SRP brings the ribosome and the whole transcription initiation complex, I used a big word, to the ribosome. And I'm sorry, to the endoplasmic reticulum, and then the polypeptide gets made inside of the endoplasmic reticulum, which can then be transported to other areas of the cell. And that's kind of what the ER does. Sometimes we refer to the ER, like in 7th grade, we might call it the highway of the cell, because it's going to allow those proteins to be transferred to other parts of the cell. All right, ladies and gentlemen, we're at the end. This is a summary image of everything that's been going on. So in transcription, there's our guitar pick. That's RNA polymerase. It made this ribbon of mRNA. We had to do all kinds of processing of it. We had to add the 5' cap. We had to add the poly A tail. We had to remove the introns and put together the exons. Remember, the exons can be put together in different ways to form different mRNAs. So they've been put together, then it leaves the nucleus, it heads to a ribosome. The trucks, the charged tRNAs bring their amino acids, and they drop off the amino acids. They bond together with peptide bonds to form our protein at the end. All right, that's it. I hope you have a good morning.